So hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CSIR PYQs of December 2023. This is shift 2 and these are part C problems that I am discussing. This is part 3 of the discussion. I will be discussing questions from biochemistry, cell biology, cell signaling, applied biology and methodology, DNA replication, protein synthesis and genetics. So coming to the first question. Given below are two figures X and Y. These are the two figures. X, Y. Representing molecular markers and parental lines P1 and P2, and F1 is the progeny. The following statement describe the nature or probable identity of markers in the above figure. Which of the following option represent combination of all correct statements? So before coming to this question, this is codominant marker and this is a dominant marker. In codominance, you will get both the bands. That is, you will get band for capital A, then small a, as well as capital A and small a in the F1 progeny. So it is a codominant marker, whereas in dominant case, you will get only single band in F1 progeny. See here, only the dominant band is shown, whereas the recessive band is not present in the case of dominant markers. Now, codominant markers, they can discriminate between homozygote and heterozygote, whereas dominant markers do not. Now coming to the question. Panel X represent a codominant marker. That is correct. You can see both the bands in the F1 progeny of capital A and small a. So panel A is X represent codominant marker is correct. Panel X represent dominant marker, that is wrong. Panel X could be SSR marker. Yes, it is a codominant marker, and panel Y could be RAPD marker. RAPD is a dominant marker. Y is showing dominant marker characteristics in the gel. Only single band is observed. And panel D is panel Y represent a dominant marker. That is also correct. So which are the correct statements here? A is correct. B is wrong. C is correct. D is correct. So A, C, D is the answer. That is option 3. Then next one. The loci for 3 mutations on X chromosome. Yellow body color, Y, cross veinless CV and forked bacills F are shown below in the map. The inference between these genes is 0. Interference is 0. That means double crossovers are taking place. A male fly with yellow body, cross veinless and forked bacills was crossed with virgin female fly homozygous for the wild type phenotype. So a male has got only X and Y and female has got 2 X chromosomes. And there will be no recombinance from male, as male has only a single X chromosome. So males will produce 50% Y, CV and F gametes and 50% will be Y gametes. Whereas the female, it will produce 8 type of gametes because recombination take place in the female. It has got 2 X chromosomes. So it will produce 2 parental gametes, 2 double crossovers, 2 single crossovers between Y and CV and 2 single crossover gametes between CV and F. Which one of the following option represent the F1 flies were, a male fly, okay, we read that. The F1 flies were SIB mated and a total of 1000 F2 progeny flies were obtained. Which one of the following option represents a correct conclusion from the analysis of F2 progeny? So you have to see how many parental types, how many single crossovers, and how many single crossovers between CV and F and double crossovers between are found. So this is the distance between Y, CV and F. So double crossover frequency is centimorgan can also be represented as percentage. So 14% into 32% into total progeny they have given as 1000. So that will come approximately 45. We will use 45 in all the other calculations. So gene distance between Y and CV, that is single crossover between Y and CV plus double crossover divided by total progeny. So 14% is equal to S single crossover between Y and CV plus 45 divided by 1000. We have to calculate for single crossover between Y and CV. That will give the answer as 95. Then gene distance between CV and F, similar to how we calculated the previous one, you can calculate here also. So that will give you around 275. Then parental type is total progeny minus total recombinants. Total recombinants are your single crossovers plus double crossovers. So total progeny is 1000. So 1000 minus 95 plus 275 plus 45, that will give you 585. And your answer is option 1. Parental type 585, single crossover between Y and CV 95, single crossover between CV and F 275, and double crossovers is 45. So that is the answer. Option 1. Then next one. Christine Newolsin Volhard and Eric Vicious carried out an extensive mutation screen to identify all genes involved in segmentation of Drosophila. The graph shows results of analyzing mutation on chromosome 2. Based on the above figure, which one of the following option is a correct statement? So these are the number of segmentation genes, that is this graph, and number of segmentation mutants, that is this dotted lines. So if you see the graph, the number of segmentation genes, they reach up till 2000 progeny analysis, and then it stabilizes. Okay, analyzing 10,000 progeny would have led to the identification of substantially more number of segmentation genes. No, by 2000 itself, till 2000 there was an increase, but after that it got stabilized. So one is not correct. Analysis of progeny beyond 2000 led to the identification of new alleles of already identified genes rather than more new genes. That is correct. More dominant mutations would have been observed in the first 1000 progeny. You cannot say whether it is a dominant or a recessive mutation. So 3 is not correct. The curves would remain the same irrespective of the mutagen that is used. Different mutagens will give different effects. So different curves will be observed. So the answer here is option 2 only. Then next one. This is a hypothetical example. The pedigree for a monogenic trait is given below. The shaded individual shows spotted skin color while the rest have uniform skin color. The individuals 1 to 9 in the pedigree were analyzed for a DNA marker, both the fragments that shows complete linkage with the skin color trait. The following statements were made regarding the above observation. Which of the following represent the combination of all correct statements? So here if you see, 1 is having the disease and it is showing 2 bands. 3 doesn't phenotypically show the disease but it has got 2 bands. Similarly, 5 also phenotypically it doesn't show the disease but it shows 2 bands in the DNA marker analysis. So this could be because of incomplete penetrance. That is why there is no representation in the phenotype, but the genotype it shows the presence of the disease. So it could be incomplete penetrance. Okay, first statement. Spotted skin color is a dominant phenotype. Yes, that is why 3 is having the disease, 5 is also having the disease even though they do not show phenotypically. So it could be a dominant phenotypes. But 3 and 5 they show the bands. So it could be a dominant phenotype. Then spotted skin color show variable expressivity. That is not correct. It is incomplete penetrance that is shown. So B is not correct. The DNA marker associated with the skin color trait is codominant. Because you can see both the normal and the defective bands. It is a codominant marker and C is also correct. The probability that the individual 5 will pass on the allele responsible for the spotted skin color to the next generation is 0.25. Now 5 it is heterozygous, it is having 2 bands. That is, it is having the diseased band and also the non-diseased band. So 50% of the gametes will be for the normal phenotype and 50% will be for the diseased phenotype. So it will pass on to the next generation is not 0.25, it is 0.5. So D is also incorrect. So the answer here is A and C only. Answer is option 2. Then next one. The figure given below is a 2 homologous chromosomes paired during meiosis where one event of recombination occurred between 2 homologs. Here is the recombination. The following interpretations were made. Which of the following option given below has all the correct answers? The individual is heterozygous for an inversion. Yes. Oh, it is heterozygous for the inversion. An inversion heterozygote is an organism with 2 homologous chromosomes. Here you have got 2 homologous chromosomes. They have already mentioned that in the question. Where one has reversed segment, that is inverted segment. This is the inverted segment and other has got the normal segment. During meiotic pairing, an inversion loop is formed so that to allow proper alignment of the homologous genes. So the individual is a heterozygous for an inversion. So A is correct. The figure depicts a paracentric inversion. So centromere is not involved. So it is a paracentric inversion. Centromere is over here. So it is not involved. So it is paracentric inversion. After recombination at anaphase 1 of 1, a dicentric and acentric chromosome will be formed. That is also correct. At anaphase 2, the recombinant chromatids will have large deletions or duplications. That is also correct. Inversions are often considered as crossover suppressors because crossover product does not survive. That is also correct. It can result in reduced fertility or offspring viability due to genetic imbalances. So E is also correct. So the answer here is A, B, C, D, E. Then next one. Consider a disease caused by a recessive allele. In a study population, 1 out of every 500 individual has the disease. Based on Hardy-Weinberg equation, what is the percentage of individuals who are carriers of the recessive allele of the disease? They have asked for carriers of recessive allele of the disease. That is 2pq you have to find out. So q square they have given as 1 by 500. Q is 0.044. Here the 0 is not visible correctly because of my overwriting. So it is 0.044 is the answer. Then p plus q is 1. P is 1 minus q. So 1 minus 0.044 is giving you 0.95. 2pq is 2 into 0.95, that will give you around 8.5%. And this is close to answer 1, that is 7.6%. So the answer here is option 1. Then next one. An ancestral sequence TTAG has diverged into 2 sequences and has since accumulated nucleotide substitutions along 2 lineages 1 and 2. This is lineage 1 and lineage 2. Match the type of substitution observed in the 2 sequences with their correct names. So you have to see whether it is multiple substitution, convergent substitution, parallel substitution, back substitution. So for that, you should know what is single substitution. So single substitution means one change, one difference. So A is giving A, there is no change. But A is giving rise to C, there is a change. So one change, one difference. So here is the change taking place and here is the difference. If it is multiple substitution, A giving C, then it will give rise to T. But 2 changes, A to C, C to T. So 2 changes are happening. A to A, no change. So 2 changes and 1 difference. So that is multiple substitution. Then there is coincidental substitution, that is A giving rise to G and A giving rise to C. There are 2 changes but only 1 difference. That is coincidental substitution. Now parallel substitution is A giving rise to C, A giving rise to C. There is no difference in the substitution, or both are C only. But there are 2 changes. So no difference, that is parallel substitution. Then convergent substitution is A giving rise to C, C giving rise to T, but A directly giving rise to T. So there is no difference in the substitution product, but there are 3 changes. 1, A to C, C to T 2 and A to T 3. Then back substitution is A giving to A, but A giving rise to C, then C giving rise to A. So there is no difference, but there are 2 changes. That is back substitution. Now in this lineage we will see which all underwent single substitution, multiple substitution, like that. So if you see CA and giving rise to T, so T is getting converted to C, C getting converted to A. But in lineage 2, it is T only. So there are 2 changes and 1 difference. 2 changes, 1 difference is multiple substitution. So 1 is multiple substitution. See here, T giving rise to C, C giving rise to A, T giving rise to T. So there are 2 changes, 1 difference, it is multiple substitution. Now see the second one, T giving rise to C, T giving rise to C. There are 2 changes. T is getting converted to C, but there is no difference between the substitution product. So it is parallel substitution. So 2 is parallel substitution. 3, A giving rise to G, G giving rise to C, A giving rise to C. So A giving directly to C, but A is also undergoing 2 changes to G then to C. But there is no difference in the substitution product. So there are three changes and no difference. It is convergent substitution. Now fourth one, if you see, G is giving rise to A, then giving rise to G. But lineage 2, it is G only. So G is getting converted to A, then to G. So there are two changes, but there is no change in the substitution product. It is G only. G has got back converted to G itself. So it is back substitution. So fourth is back substitution.